Okay. Well, first, I would like to thank the organizers for giving me the opportunity to present some of the work here. So I will talk about calcium signals, and the first slide I'm showing is I'm showing it not because Philippe is in the audience. I'm showing it even when he's not around. It just shows you what everybody who works with Philippe should show. That it's a human biopsy of a patient. It's a view of a human biopsy of a patient in acute shigellosis phase. We could clearly see the infiltration of polynuclear cells that promote the inflammation and the destruction of the microvilli linked after Shigella infection. So as you know, Shigella is an enteropathogen that invades the colonic epithelium, and it has the ability to have a pointer here. So. Okay, so it can invade epithelial cells, it can lyse the phagocytic vacuole, escape, and while it replicates, it induces many processes that it diverts through a specialized secretion apparatus that we'll talk about. And during this multiplication phase, it can polymerize actin to spread from cell to cell, and thereby disseminating without the entire epithelium, without extracellular steps. So this movie here is just a regular spinning confocal microscopy of Shigella invading cells. And you can see that upon invasion, the bacteria is inducing the membrane ruffling that is here rich in actin. And the process takes about 10-15 minutes. So the bacteria enters, contacts the cells, triggers actin polymerization that leads to membrane ruffling, and is engulfed in a large micropanocytic vacuole that he lies outwards. So this slide, I won't go into the details, just to tell you that we know quite a bit about the various effectors, and that's been the work of about, I would say, 20 years, that allows bacterial invasion. We know signaling pathways, we know targets, and what I would like to emphasize is that most of this work concern proteins and cellular targets proteins as well. It's because this is easy to test. We can mutate genes, and we can, of course, try to look at the function easily and make a relationship, a functional relationship with the effectors. What I would like to talk about is something that people don't usually look at, and this is, I think it's in line with the seeing is believing focus of this topic, because here we're looking, okay, of course, to believe you need to see, but to see you need to look. And of course, it depends. There are things that you don't see if you don't look for, right? So this is exactly the case for calcium. Calcium is there all over the place. It's very important because it regulates the function of essential proteins. It has to play a role because we know that calcium concentration can vary quite a bit. This is just a slide to show you the regulation of calcium hemostasis. Most of you know, of course, that calcium concentration is very low in the cytosol of host cells, about 100 nanomolar. But upon certain stimulations, such as agonist stimulation, you would have hydrolysis of PIP2, PI45P2, by PLCs that would generate IP3. And this IP3 will in turn allow the release of calcium from pools, and will switch, which is in the order of millimolar range, and which in very drastic way impact on the concentration of calcium. So with the advent of calcium imaging technique, and here I'm referring to the advent of fluorescent probes that allow single-cell imaging, and we know much more about calcium imaging. So fluorescent probes, of course, are the ones that have been developed by Roger Chin that owe him the Nobel Prize, among other things, and I don't believe that people give the Nobel Prize to anybody. So what they're able to see here is that the calcium signals actually can be decomposed in various macro signals. So first, one could image the opening of elementary channels, what they call blips, that would lead the release of a minute amount of calcium molecules, and that one can image because that lasts only for 100 milliseconds. Of course, the duration of those response is linked to the diffusion of calcium within the cytosol, which is very fast. Then if you have a cluster of those channels that release from the pools more calcium, we have what is called puffs, and those are a bit larger. They can last 400 milliseconds. And then eventually those responses will integrate to above a threshold that would trigger a whole global calcium response that would lead to the release of calcium from the whole pool of the cells. And I'm saying that because it's important, I believe, again, for the function of proteins. It's known, of course, that those responses are of course regulated because calcium cannot stay high in a cytosol for a long time, otherwise it would be toxic for the cells. So therefore, very often, at least under physiological conditions, the calcium concentration raises and decreases and is very transient. And very often it oscillates. And the frequency, for example, of oscillation depends on the concentration of agonist. And it's been shown, for example, that there's a calcium code, meaning that the cell is able to decode the frequency and the type of signal that, that is generated. For example, this oscillatory pattern would lead to activation of transcriptional factor, NF-kappa B, and inflammation, while sustained calcium response would lead to NFAT and cell death pathways. So about 10 years ago, when we started to look at that with Laurent Combet in Orsay, we noticed that Shigella, during invasion of epithelial cells, is able to induce global calcium response, but he was already struck by the shape of those response. If one looks at agonists here, so this is agonist-like, it doesn't matter, one could see that the calcium response induced by Shigella has very slow dynamics. It raises slowly and decreases slowly, and it's often isolated with no oscillatory pattern. And we got stuck there. More recently, we went into looking at local calcium response. And what I'm showing here is just a very fast imaging of calcium. This is like one acquisition every 30 milliseconds. And this is here the same field, but with phase contrast, where we can clearly see the entry site where the bacteria enters the cell. And you can clearly see that where the bacteria enters, we have a local calcium response, I hope you can see it, that increase. And this response is quite atypical because I told you that local calcium response by nature are very transient. What we're able to determine there is that at the site of invasion, the local calcium response could last up to several seconds, which is highly atypical. So first, of course, we thought that it was important to try to understand why those calcium responses were local and were so atypical, because through other experiments, we had some hints that local calcium response were important for actin polymerization at entry sites. So first, to explain how those calcium response could stay for so long at the same spot, we first made the hypothesis that there was a local perturbation of calcium hemostasis at the site of Shigella invasion. So in other words, as opposed to agonist, when you have a stimulation throughout the cell surface, bacterial invasion stimulates PLCs and IP3 production locally at one site. And it's possible that because the stimulation is local, you might have a local production of IP3 there that would explain the calcium response. And this is because, as you know, there's not a linear relationship between the IP3 concentration and calcium release. In other words, for low levels of IP3, you can have high levels of calcium. So what we did first is just to stain for IP3 receptors. So IP3 receptors, as you know, all those receptors are present in the ER membrane that upon IP3 binding would open and release calcium in the cytosol. And we could clearly see that at entry site, we have a very strong enrichment of type 1 and type 3 IP3 receptors. Those are the receptors that are expressed in Hill cells. And if one quantifies, it's about a twofold enrichment compared to the rest of the cell. But when we try to model, and then we collaborate with this, people expert in calcium imaging and modeling, we could not explain those long-lasting response only through the recruitment of IP3 receptors that increase IP3 levels. We thought that there was other parameters that could explain the local restriction of calcium signals, and to cut a long story short, we just wanted to FRAP experiments, so fluorescence recovery after photobleaching experiments, to try to assess the level of diffusion at Shigella invasion sites and to see whether diffusion, the restriction of diffusion could affect the confinement of those response. And what we could see here through those experiments is that indeed, in control area, for example, we have like a half-time recovery of about 145 milliseconds. In entry foci, you have almost 1.5 times more restriction and diffusion, which is clearly indicating there's a strong restriction diffusion is linked to actin polymerization in that case, because we can go back to control level if we treat the cells with cytokeratin D. So in our simulation, when we went back, then we can clearly show that the local calcium response induced by the bacteria stays high for a long time because there's more IP3 recruited entry sites on the one hand, and on the other hand, because restriction is limited. And if you affect one or the other of those parameters, then you cannot explain those local restriction of those local calcium signals at the very long entry sites. So I was talking about the role of effectors during invasion and the fact that people do not look only at other things like calcium. So I just want to, as a proof of concept, to try to check if other bacterial effectors that are secreted upon cell contact might have a different activity depending on calcium. So this is a protein called IPGD. So IPGD is one of those proteins that is injected in the bacterial cytosol, and you heard about the type 3 secretion apparatus from at least 2 speakers this morning. IPGD is a PI45P2 phosphatase, so it hydrolyzes PIP2 into PI5P, and this has been shown that PI5P, it has been shown to have 2 roles. One type of set of roles that is linked to PI5P production, and this would be, for example, the activation of the AKT PI3 kinase pathways, or the sequestering of EGF receptor in early endosomes. And other activities linked to PI45P2 hydrolysis, and this would be, for example, de novo actin polymerization, because it would disconnect actin filaments from the cell membrane, or the inhibition of T-cell migration through ezrin, through the dephosphorylation of PIP2 on ezrin molecules. What we hypothesize is that IPGD could probably also affect PIP2. And again, this slide you show, but if you have here IPGD, hydrolysis of PIP2 would deplete the cells from the substrate of PLC, or that PLC usually used to produce IP3. So the prediction is that IPGD hydrolysis would decrease PIP2 levels and then decrease calcium signals linked to IP3 signals. And we knew that Shigella induced calcium signals are dependent on IP3. So we did the same thing. We infect cells with the IPGD mutant, and what you see here is just a movie showing the typical examples of what we can see in the case of this mutant. So if you just focus for the time being on this panel here, the white arrow indicates where the entry site occurs. And you can clearly see that in that particular case, we have a local calcium response that oscillates, but it's far away from the entry site. And this would be reminiscent of oscillating puff occurring at a distance from the bacterial invasion site. And the other example is just global response that we can see very often with this bacteria. So as opposed to the wild type strain that induces mostly, and this is the same view but this time quantified, this is in the area of different areas of the same cells. Here in the case of the wild type, you can see the calcium signals in the case of the wild type are local and they remain confined in entry sites. Whereas in the case of the IPGD mutant, you very often have like global responses of calcium or puff-like oscillating responses that occur at a distance from invasion site. And if one quantify, one can clearly see that the wild-type Shigella induces more confined local response, whereas the IPGD mutant induces mostly global responses. So what is the incidence of that? Where if one looks at longer kinetics of infection, one could see that one can say that again, in the case of the wild-type Shigella, you can see responses are isolated and with very slow dynamics, and eventually those responses die over time. And if you stimulate the cells with calcium agonists, then the cell is unable or very inhibited in its ability to elicit calcium responses. In contrast, if one look at IPGD mutant infected cells, one can clearly see response that oscillates or are sharper. And if you stimulate cells with agonists, then the cell is still able to induce calcium response. So all this is consistent with the notion that IPGD acts as a calcium inhibitor or modulator of calcium response. It allows the bacteria to confine calcium signals at the site of invasion, and over prolonged kinetics, it would inhibit calcium response. So to explain this difference, again, we went over the same type of experiment that we did with when we first tried to explain local response, a long-lasting local response induced by wild-type Shigella. So here we went through quantification of IP3, so through GFP-pH labeling, or IP3 receptor. And in both cases, one could see that there's more IP3 receptor and more IP3 at invasion site, which is consistent with the notion that IPG hydrolyzes PIP2 and therefore depletes the cells from substrate for IP3 production. And we also looked at diffusion restriction, and we could see that the IPGD mutant is also less restrictive in diffusion than the wild-type foci. And we had this hint before because just looking at entry site, we saw that the IPGD mutant made foci where actin was less dense, less polymerized, and therefore this is consistent with the notion that the IPGD mutant induces foci that are less restrictive in diffusion. And if one combines the two notions, then one can understand why IPGD confine local signals. So this is, again, a cartoon with a pseudocolor explaining here the calcium concentration. In the case of wild-type Shigella, you would have IPGD that hydrolyzes PIP2 and then depletes cells substrate for PLC. So therefore, you have IP3, but just at the right level, so that the signal stays confined at entry sites, and very little goes to stimulate global responses, most of it going towards PI5P production. But then when IPGD is not around, what you have is a lot of PIP2 being recruited and hydrolyzed, IP3 production that stimulates global responses, and then responses that keep going over in the infection time. And what happens over infection? Okay, of course, you might wonder why the cells do that. Why does it want to confine calcium responses? So we believe that probably this modulation at entry site is also important for membrane processes, like protein involved in cytoskeletal organization. But here we looked at long-term prolonged kinetics of infection. And what we could see here is that after this is 4 hours of infection, one can see that cells here stained in red for actin and green for bacteria are still remarkably spread for the amount of bacteria they contain. In contrast, when you look at the IPGD mutant, you can see at this period, the cells already are round up and detached very easily, indicating that the IPGD mutant is more cytotoxic than wild-type Shigella. And if one quantify that, you can see that this cytotoxicity is quite significant because it can be up to here threefold, it can be up to fivefold, depending on the experiments. And this is here wild type, this is the IPGD mutant, this is just control. And the cytotoxicity is calcium dependent because if you remove calcium from the medium, then you have cytotoxicity that is no different between wild type and the IPGD mutant. So how does that work? So we think, that this is calcium dependent, and we know that we had the hint that the cells in the IPGD mutant will round up and lose its adhesive structures rapidly. And we know that there are some proteases, like for example, disassembly of focal adhesion involves the activation of the calcium-dependent protease calpain, which will cleave focal adhesion proteins and disassemble focal adhesion. So we looked at calpain activation, and this is just standard blots looking at the disappearance of calpain, because calpain, upon activation, is autoproteolized, so therefore it disappears. And you can clearly see that in the IPGD mutant, this cleavage occurs more rapidly than in the wild type, and this is quantified here. Sorry. And so one of the targets of calpain as a focal adhesion protein is tallin. So if one looks at tallin, and this is, again, cells transfected with tallin GFP this time, infected with Shigella, we can see that after 2 hours, you have cells that are very well spread that form those very nice focal adhesion, even though they contain a lot of bacteria. In the case of the IPGD mutant, you can clearly see a disappearance of those focal adhesion, and this is quantified here. You can clearly see that the IPGD mutant here in black loses its focal adhesion much more rapidly than wild type. And if one goes the other way around, the appearance of cells with focal adhesion, you can clearly see the reverse pictures, with more cells showing no focal adhesion compared to in the IPGD mutant compared to wild type. This is just control here. So to summarize what I've told you, so what I showed you is that Shigella induces a typical long-lasting calcium macrodomain at evasion sites, and we truly believe that this is a key factor that regulates not only the host cell function, but perhaps also bacterial effector function. That IPGD favors the elicitation of local sustained calcium responses by confining those responses at entry sites, and prevents global calcium response during Shigella invasion. So this is particularly obvious when you look at long kinetics of infection, where you still see calcium signals in cells infected with the IPGD mutant. We've shown that IPGD prevents cytotoxicity during Shigella infection, and this is in a calcium-dependent manner, which is in line with its effect on calcium signals inhibition. That it downregulates, therefore, calpain activation during Shigella infection. It depends on extracellular calcium. And that one of the targets is taline, the focal adhesion protein, that is cleaved by calpain, and that is degraded differentially in the wild-type and IPGD mutant. And we believe that IPGD prevents calcium-dependent cell death linked to the loss of adhesion detachment. And this is probably important to do that because, and this is what we realize, we know that eventually the cell will die when infected with Shigella, but it makes a big difference whether it dies quickly or slowly. And this is probably very important for the bacteria to set up its program to disseminate from one cell to the other and to efficiently colonize the epithelium. And for that, I'll just let people do the work. So the work of the people who did the calcium in my lab are Chen Sunhui, who will defend his PhD thesis next fall. Kevin Denny did his internship last year in the lab, and this work was done in collaboration with the lab of Philippe Sansonetti and with Christopher Nhieu in Belgium, as well as with the group of Laurent Combet in Orsay, and the Geneviève Dupont for the mathematical modeling in Brussels. Thank you.
Thank you, Guy. Are there any questions? Yes. Okay. Joost?
Thank you, Guy. I very much like the talk. One of the most striking features of IPGD, I think, is probably the induction of macropinosomes at the entry site. And I'm just wondering, because we've been sharing that data. Where does the calcium come from? Could it be that the macropinosomes play a role there?
Okay, so what we know is that the calcium signals are IP3 dependent, so they have to come from intracellular pools, at least from the beginning. It's possible that afterwards the influx plays a role, but you don't need the influx to have those local, long-lasting calcium responses. Perhaps for the maintenance of those, of those macrodomains, you need the influx, but not for the initiation at least. And that would suggest that somehow, there's the component of intracellular pool that is required, and that would be consistent with the recruitment of STIM, the STIM protein at entry site that we observe.
So there's been descriptions of specific sites in which the ER comes into contact with the plasma membrane, and I just wondered whether they may be playing a role in your local calcium release, and have you looked at this?
Yeah, so it's exactly what I tried. So we looked at STIM. So STIM is this calcium sensor that senses the luminal levels of calcium within the ER, and that's been described by many people to dock to the RA channels at the plasma membrane upon depletion of the pools. And when we look at that, we see recruitment of STIM1 as well as RO1 at entry site, but it seems that the siRNA doesn't do much. So it's not so clear that, at least in terms of calcium, those play a role there.
I had a question, actually. So you kind of hinted at it at the